PharmaPal.
You're listening to Bloomscast, the official podcast of Blooms.io, home of the bioactive library for mass spectrometry instant optimization. Let's dive into the latest in natural chemistry, bioactive discovery, and the future of lab innovation.
Today, we're zeroing in on Shikonin, that really striking red pigment you find in the roots of plants like Lithospermum erythrorhizon.
Yes, Siko in traditional Chinese medicine.
Right. And it's also in, well, a whole family of plants, the Boraginaceae family.
Exactly. And, you know, while its color is great for dyes and cosmetics, historically,
It's a visual appeal.
the real story, I think, is its medical potential. It's pretty impressive. We're talking reported anti-cancer, anti-thrombotic, neuroprotective, anti-diabetic, antiviral,
Wow. Okay, hold on.
anti-inflammatory, antioxidant, antimicrobial properties. It's quite the list.
It really is a list. It sounds like some kind of botanical Swiss Army knife. So our mission today is, well, let's cut through the hype a bit and really explore the science.
Sounds good.
Based on the research you shared, we want to look at where it comes from in nature, how it actually seems to work,
Mechanisms, yeah.
the clever ways scientists are trying to maybe make more of it or make it in the lab, and, you know, what's next for Shikonin and its variations.
Right. Lots to cover.
Definitely. So let's start right at the source. Where do we find these plants?
Okay, so the primary source, the main one, is the dried roots of Lithospermum erythrorhizon. As you said, long history in traditional Chinese medicine. But that whole Boraginaceae family is pretty interesting. You've got several others that make Shikonin or, you know, very similar compounds.
Like which ones?
Well, names like Ichium licoris, Arnebia pop up, and even Alkanna tinctoria. That's where we get the name Alkannin from, which is Shikonin's close relative.
Oh, okay. Alkannin. We'll probably come back to that. So it's this whole family. Where are they mostly found, geographically speaking?
They're actually pretty widespread, distributed globally, but there does seem to be a significant concentration in Asia,
Okay.
particularly the northwestern Himalayas. So yeah, while you might find them elsewhere, that region seems to be a real hotspot.
Got it. Himalayas, key area. Now, you mentioned Alkannin. The research talks about acetylshikonin and this idea of an AS chiral pair. Can you unpack that a bit?
Sure. So acetylshikonin was, I think, the first specific derivative isolated from Lithospermum.
Okay.
Now, the AS pair that refers to Alkannin and Shikonin and S, they're enantiomers,
Meaning
they're like mirror images of each other. Think of your left and right hand, same parts, but arranged differently in 3D space.
Right, chirality.
Exactly. And what's really fascinating is that this slight difference in shape, this handedness, can actually lead to different, sometimes even opposite biological effects.
Opposite. Seriously?
Yeah, potentially. Like one might be better against certain microbes, or have stronger anti-cancer activity, or affect wound healing differently than its mirror image.
Wow. So that tiny structural twist makes a huge functional difference.
It can, yes.
Yeah.
For today, though, we're mostly focusing on Shikonin itself, which is the R enantiomer, so one specific hand.
Okay.
But it's really important to remember that this chirality exists and matters. And the research also points out there's a whole bunch of Shikonin derivatives, different variations on the basic structure.
Right. Suggesting even more possibilities. Okay, so we know where it comes from, the basic chemistry. How does the plant actually make it? What's the biosynthesis like?
Okay, let's try a simplified view. Imagine like a tiny factory in the roots, and you've got specialized workers. These are enzymes. They take simple starting materials and step by step transform them into Shikonin.
Like an assembly line.
Pretty much. Key enzymes involved have names like CYP76B101, CYP76B74. These are types of enzymes that often add oxygen atoms in very specific places.
Okay, specific tools for specific jobs.
Exactly. And there are others too, like polyphenol oxidase. And surprisingly, some research found enzymes similar to ones involved in making cannabinoids in cannabis plants.
Really? That's unexpected.
Isn't it? And then you have a group called BAHD acyltransferases, specifically LESAT1 and LEC1 found in Lithospermum.
And what do?
Think of them as decorators. They attach different little side chains, acyl groups, like acetyl, isobutyl, onto the main Shikonin or Alkannin molecule.
Ah, creating those derivatives you mentioned.
Precisely. LESAT1, for example, tends to work on Shikonin, adding these groups. LAT1 does similar things, maybe preferring Alkannin or different groups. That's how you get the diversity.
It sounds incredibly precise, that whole pathway. And I guess given how useful Shikonin might be, people are trying to boost that production?
Oh, absolutely. Researchers are trying several clever ways. One is genetic engineering.
How so?
Well, for instance, they took a gene called UBA from E. coli, the common bacteria, and put it into Lithospermum erythrorhizon.
Yeah.
And it significantly increased Shikonin production.
Wow. So borrowing a gene from bacteria can turbocharge the plant's own factory?
That's the idea.
Yeah.
Another technique involves using specific bacteria like Agrobacterium rhizogenes to induce what they call hairy roots.
Hairy roots? Sounds interesting.
It does. But these modified roots often produce much higher amounts of Shikonin derivatives.
Okay.
They're also trying things like boosting the plant's own genes. Overexpressing a gene called FGHQH1 in a related plant, E. plantagineum, led to more acetylshikonin.
So tweaking the plant's genetics or using these hairy roots? Any other tricks?
Yeah, it's not just the plant itself. Sometimes introducing beneficial bacteria into the hairy root cultures, like with Alkanna tinctoria, has also boosted yields.
So sort of creating a helpful little ecosystem for the roots.
Kind of, yeah. And then there are environmental factors.
Light and water.
Exactly. Adding L-phenylalanine, which is a building block for Shikonin, to the growth medium can help.
Makes sense. Feed the factory.
Right. And interestingly, darkness actually seems to increase Shikonin content in some cases. Light might decrease it.
Huh. Counterintuitive, maybe.
It can be. And pH, the acidity or alkalinity, is also key. For some species, like Arnebia euchroma, a slightly alkaline pH around 8.75 seems optimal. Maybe helps the enzymes work better.
Fascinating. So many levers you can potentially pull inside and outside the plant. Now, plants are one source, but what about making it completely synthetically in a lab?
Yes, that's another important avenue, chemical synthesis. It gives you more control, potentially. Several methods have been developed for the total synthesis.
Wait. Well, one of the early ones was by Terada and colleagues back in the 80s. More recently, you've got approaches like Luth et al.'s method using specific naphthalene starting materials.
Okay.
And Wang et al. Developed a route using a ruthenium catalyst. What was neat about that one was they achieved very high enantiomeric excess.
Getting mostly the correct handedness, the right mirror image we talked about?
Exactly. High purity of the desired Shikonin enantiomer, which, as we discussed, can be really important for biological activity.
Right. So lab synthesis offers an alternative, maybe more controlled route. Okay, let's switch gears to the really exciting part. What does it do? Those pharmacological activities. Where should we start?
Let's kick off with anti-inflammatory effects. That's a big one.
Okay.
Animal studies, like the classic rat paw edema model,
Right.
where they induce swelling,
Right.
have shown that Shikonin and its derivatives, especially acetylshikonin, can significantly reduce that inflammation. Acetylshikonin seems particularly potent.
And how is it working? What's the mechanism for reducing inflammation? It seems to act in a few ways. It can interfere with allergic reactions by affecting how calcium moves in cells. It also seems to block a receptor called MRGPRX2, which plays a role in inflammation and itch.
Okay, multiple angles.
Yeah. There's also evidence it might help with neuroinflammation in the brain and potentially protect the liver from damage during sepsis.
Wow.
And one study even suggested it helps heal lung injury by regulating specific microRNAs and a receptor called toll-like receptor 4. So it's hitting various points in that complex inflammatory process.
Really hitting it from different sides. Okay, what about antimicrobial effects? You mentioned a broad range earlier.
It's quite broad. Studies show activity against bacteria involved in gum disease, like Porphyromonas gingivalis and Fusobacterium nucleatum.
Oral health.
Yeah, and common ones like Staph aureus, E. coli, strep pneumonia. Also effective against the fungus Candida albicans.
So bacteria and fungi.
Correct. And interestingly, some recent computational studies, docking studies, suggest it might bind to the main protease of the SARS-CoV-2 virus.
The COVID virus.
Potentially, yeah. Suggesting it could interfere with its replication. Needs more research, of course, but it's an interesting lead.
Definitely.
And people are also embedding Shikonin or Alkannin into nanofibers, like tiny threads, which then show antimicrobial activity. Could be useful for wound dressings, perhaps.
That is clever. Okay, let's move to the area that seems to get a lot of attention. Anti-cancer effects. What's the story there?
Yeah, this is probably one of the most heavily researched areas for Shikonin. And it seems to attack cancer cells through a whole host of mechanisms.
Like what?
Okay, so it can inhibit proliferation,
Right.
stop the cells from multiplying, It can induce apoptosis, basically, tell the cancer cells to self-destruct.
Right, programmed cell death.
Exactly. It can arrest the cell cycle at various points, stopping division. It interferes with tubulin polymerization.
That scaffolding thing again?
Yeah, messing up the cell's internal structure needed for division. It also impacts major signaling pathways that cancer cells often hijack, like PI3K cantiMTOR, beta-catenin, STAT3.
So disrupting their communication networks.
Pretty much. It can also increase levels of PAHP, which maybe paradoxically can lead to DNA damage in cancer cells. It inhibits key energy-producing enzymes like pyruvate kinase M2 and growth factor receptors like EGFR. It can even turn down cancer-promoting genes like CMYC.
That is an incredible number of different ways to fight cancer cells. It's not just one target.
Not at all. It seems multifaceted. And it's shown activity against a wide range of cancer cell lines in the lab.
Which ones?
Oh, HeLa cells, U937 leukemia cells, various breast cancer lines, AKI-49 lung cancer, HE29 colon cancer, melanoma cells, many different types.
And are people looking at using it with other treatments?
Yes, there's evidence of synergy. Combining Shikonin with things like melatonin or even silver nanoparticles seems to boost the anti-cancer effect in some studies.
Combination therapy potential.
Exactly. And researchers are also designing new molecules based on Shikonin, like those Shikonin benzofuran derivatives you mentioned, trying to improve activity and maybe reduce toxicity.
Very promising. Okay, moving beyond inflammation and cancer, antioxidant effects. Why is that important?
Well, antioxidants combat oxidative stress, which is caused by unstable molecules called free radicals.
Right, those damaged cells.
They do. They contribute to aging and potentially to diseases like Alzheimer's. Shikonin has shown it can scavenge these free radicals. It can also inhibit lipid peroxidation damage to fats in cell membranes and chelate metals that can promote harmful reactions.
So it's protective against that kind of underlying cellular damage.
That seems to be the idea, yes.
Yeah.
Suggesting potential in age-related conditions or neurodegenerative diseases.
Interesting. What about diabetes and obesity? Any potential there?
There's some intriguing evidence. Acetylchikonin, for example, has been shown to improve glucose uptake in muscle cells.
Which would help lower blood sugar.
Potentially, yes. Also, extracts with Shikonin can inhibit enzymes like alpha amylase, which digests starch, and pancreatic lipase, which digests fat.
So affecting digestion of carbs and fats.
Right, suggesting possible roles in managing blood sugar and maybe weight. Some animal studies have actually shown Shikonin helping to reduce body weight and blood lipid levels.
Okay, another area to watch. Now, what about wound healing? You mentioned its traditional use. Is that backed up?
It seems to be. Traditionally used for burns and skin issues is well documented.
Right.
And modern studies, for instance with Alkanna tinctoria extracts, show they promote faster epithelialization, new skin growth, and angiogenesis, formation of new blood vessels, both critical for healing.
So science confirms the traditional wisdom.
In this case, yes. And there's that nanofiber research too. Scaffolds containing Lithospermum extract seem to promote wound healing in studies.
Okay. And just briefly, neuroprotective effects. We touched on neuroinflammation.
Yeah, it ties back to the anti-inflammatory and antioxidant actions. The idea is that by reducing inflammation and oxidative stress in the brain, it might help protect against conditions like Alzheimer's. It's an active area of research.
Got it. And the last couple from that initial list about antithrombotic, antifibrotic.
Right. It's been shown to inhibit something called PAI1, which is involved in blood clotting. So that suggests an antithrombotic, anti-clotting effect. It can also interfere with how certain immune cells stick to blood vessel walls, which is relevant in atherosclerosis. The antifibrotic effects are perhaps less explored, but might relate to tissue scarring processes. Anti-gonadotropic effects seem less studied recently compared to these other areas.
It's just such a wide range of activities.
Yeah.
Amazing. But with all these potential benefits, what about safety, toxicity, side effects?
That's always the critical question. Animal studies, like in rats and beagle dogs, have generally suggested it's relatively safe at certain doses. They didn't see major adverse effects.
Good.
However, and this is important, in vitro studies, lab dish studies show Shikonin can inhibit several cytochrome P450 enzymes.
The liver enzymes that break down drugs.
Exactly.
Yeah.
That raises a red flag for potential drug-drug interactions.
Yeah.
If you took Shikonin with other medications processed by those same enzymes, it could cause problems.
Right, altering the levels of other drugs. That needs careful study.
Definitely. On the plus side, some of those synthetic derivatives we talked about, some have actually shown lower toxicity in lab tests than Shikonin itself, or even standard drugs they were compared against.
So maybe modifying the molecule could improve the safety profile too. Interesting. Okay, so we have all this lab research, some animal data. Is any of this translating to humans? Patents, clinical trials.
Yes, it is starting to. There are quite a few patents filed around the world related to Shikonin and its derivatives.
For what kind of uses?
A lot focus on dermatological applications, skin conditions, which makes sense given the traditional use and wound healing data. Also, quite a few related to anti-cancer applications.
And clinical trials, are people actually testing it?
If you search databases like clinicaltrials.gov, you do find some. For example, trials looking at Lithospermum erythrorhizon extracts for conditions like rhinitis, nasal inflammation, and even uterine fibroids.
Okay, actual human studies.
Yes. And there was mention of a study on a traditional Chinese medicine decoction containing Lithospermum for shrinking tumors. Plus, patents exist just for better ways to extract Shikonin and Alkannin from the plants.
So, real movement towards application. But you mentioned earlier, it doesn't dissolve well in water. That sounds like a challenge for making it into a medicine.
It's a major challenge. Core water solubility and sometimes stability issues can really limit how well it gets absorbed, how much actually reaches the target in the body. Bioavailability issues.
Right. So how are researchers tackling that?
Nanotechnology is proving really key here.
Tiny particles.
Exactly. Developing nano delivery systems. Things like liposomes, which are tiny fat bubbles, or microemulsions, or solid nanoparticles. You encapsulate the Shikonin inside these carriers.
And that helps how?
It can dramatically improve solubility, protect the Shikonin from degrading too quickly, enhance absorption, and even allow for targeted delivery.
Targeted delivery. Like sending it straight to a tumor.
That's the goal. Designing nanoparticles that specifically accumulate in tumor tissue, or even cross the blood-brain barrier to treat brain cancers like glioma, that's a huge hurdle nanotechnology might help overcome.
Wow.
They're also looking at co-delivery, putting Shikonin and another drug together in the same nanoparticle to get synergistic effects right where they're needed.
So nano is really helping bridge that gap from potential to practical use.
It seems absolutely crucial for unlocking Shikonin's full therapeutic potential. Yeah. Tackling those fundamental delivery problems.
Well, this has definitely been a deep dive. We've covered a lot of ground from its natural sources in these fascinating plants to its really quite remarkable range of potential medical benefits, especially that anti-cancer and anti-inflammatory activity.
Which seem particularly strong, yeah.
The different ways scientists are producing it, synthesizing it, and now using nanotechnology and chemical tweaks to make it work better.
We really have hit on some key points. That whole enantiomer thing, Shikonin versus Alkannin having different effects, that's fascinating.
It really is.
And just the complexity of how the plant makes it, the biosynthesis, plus the sheer number of ways it seems to interfere with cancer cells. That's not just one trick.
Absolutely. And it's encouraging to see the patents and the clinical trials starting up, suggesting real translational potential. But as you said, still more work needed, especially on those potential drug interactions.
Definitely. Which kind of leads to a final thought, perhaps, for you listening. Given everything we've discussed, this incredibly broad range of activities, the advances in targeted delivery, could Shikonin, this natural pigment, actually become a cornerstone therapy in the future for multiple diseases, really moving beyond just its traditional roots? And, what other potential applications might its unique properties unlock that we haven't even thought of yet?
Thanks for tuning in to Bloom's Cast, brought to you by Blooms.io. Stay curious, stay connected, and we'll see you next time on the frontier of natural product innovation.
PharmaPal.